Welcome to the Deep Dive, where we break down complex molecular processes into, well, insights you can actually use. Okay, let's unpack this. Today, we're diving into the pretty complex world of how eukaryotes and archaea handle their genomes. And we're going to look at it through a very real-world lens, plastic pollution. Yeah, it's staggering. Globally, something like 140 million tons of plastics, polyurethane, polystyrene, you name it, get bought and thrown out every year. And making it takes, what, 150 million tons of fossil fuels? These things stick around for ages.
But here's where biology offers maybe a bit of leverage. There are microbes out there, like the bacterium Ralstonia eupropha, that naturally make and break down similar polymers. They're called PHAs, polyhydroxyalkanoids.
DHA, so basically bioplastics.
Exactly. They're natural bioplastics. The bacteria make them inside their cells as a way to store carbon, like little energy reserves.
Okay, so we have a microbe that makes bioplastics. That sounds like a ready-made solution. Why isn't this happening everywhere? What's the catch?
The catch is usually cost. Our eutropha is what we call a chemoorganoheterotroph.
Chemo.organo.heterotroph. Okay, break that down.
It just means it needs to eat organic molecules to live and grow. You have to feed it things like sugars or organic acids to get it to make those PHAs. And those food sources, they can be expensive.
Right. So even if it can make a lot of PHA, you said like 80% of its weight, feeding it costs too much for large-scale production to make economic sense.
Precisely. So the clever idea became, what if we take the genes for making PHAs from the bacteria and put them into an organism that, well, feeds itself?
Ah, like plants. I think I read about attempts with tobacco or Arabidopsis.
They tried that, yeah. But using crop plants raises other issues, land use, competition with food crops. It gets complicated and still potentially expensive. So researchers kind of pivoted back to microbes.
But a different kind of microbe.
Right. A proof of concept involves putting those bacterial genes into a microalga, specifically a diatom called Phaeodactylum tricornutum.
The diatoms, that's algae. Like pond scum.
Sort of. Yeah, it's a type of single-celled algae. And the key thing is it's a photoautotroph. It just needs sunlight, water, and CO2. You can grow it in places you wouldn't grow crops. Much cheaper feedstock.
Okay, that makes sense. So the goal is to get this diatom, this eukaryotic organism, to express those bacterial genes really well.
That's the challenge. And that brings us right to the core of our deep dive today. To get maximum expression in a eukaryote, you absolutely have to understand the fundamental differences in how eukaryotes process their genetic information compared to bacteria.
And where do archaea fit into this picture?
Ah, Archaea. They're the fascinating twist. Carl Woiss's work showed us that even though Archaea look like bacteria under a microscope and live in similar ways, their machinery for handling DNA and RNA replication, transcription, translation is actually much more similar to eukaryotes.
So they're like a hybrid system.
In a way, yes. They're this incredible evolutionary mix, which makes studying them vital if we want to bridge that gap between bacterial genes and a eukaryotic host like our diatom.
Okay, let's dig into those differences, starting with copying the DNA replication. Bacterial replication seems straightforward, right? Circular chromosome, one starting point, one main enzyme, DNA pol 3 does most of the work.
Pretty much, yeah. It's efficient, elegant in its simplicity. But eukaryotes, they had, well, bigger problems. Literally.
Bigger genomes, linear chromosomes.
Exactly. Much, much larger genomes. DNA split across multiple linear chromosomes, and all that DNA is wrapped tightly around proteins called histones, forming structures called nucleosomes. You can't just start at one point and hope to finish copying in time for cell division.
So they needed a way to speed things up, parallel processing.
That's the key. Multiple origins of replication scattered along each chromosome. Each origin kicks off replication, creating these independent copying units called replicons. Think of yeast chromosome the third. It alone has 9 origins.
And starting that process must be more complex, too.
Oh, definitely. It begins with a protein complex called ORC, the origin recognition complex. It acts like a landing pad, marking the start. Then it recruits other proteins, including helicases that unwind the DNA, getting everything ready for their replication machinery, the replisome.
And that replisome itself is different, not just one main polymerase.
Yep. Eukaryotes use a team of specialized DNA polymerases. Pol alpha primus gets things started by making a short primer, a little bit of RNA, a little bit of DNA.
Okay, the starter block.
Right. Then Pol epsilon takes over synthesis on the leading strand, the one that's copied continuously, and Pol delta handles the lagging strand, the one made in pieces. This division of labor is crucial for handling the complexity.
It's a whole different level of organization. But maybe the biggest hurdle wasn't just size, but the shape. Those linear ends, what happens there?
Yes. Here's where it gets really interesting. That's the famous end replication problem. Because DNA polymerases need that primer to start, they can't copy the very, very tip of the lagging strand.
So every time the cell divides, the chromosomes get a tiny bit shorter.
Exactly. Those ends, called telomeres, would just erode away. And shortened, unprotected ends are dangerous. The cell might see them as broken DNA, leading to fusions or degradation. It's like a ticking clock.
So eukaryotes evolved telomeres as protective caps, and this enzyme, telomerase, to basically rebuild them.
Precisely. Telomerase is ingenious. It's a type of enzyme called a reverse transcriptase, meaning it can make DNA from an RNA template. And the cool part, it carries its own RNA template inside itself.
Wait, it brings its own instructions.
It does. This internal RNA template lines up with the repeating sequence at the telomere ends, allowing telomerase to add more repeats, extending the DNA and counteracting that shortening. It keeps the chromosomes stable.
Okay, that's a pretty sophisticated solution. Now back to Archaea. Where do they land? You said circular genomes like bacteria?
Right, structurally simpler in that sense, no end replication problem. But the replication machinery, the proteins like the MCM, helicase, the primase, these are clearly relatives of the eukaryotic versions, not the bacterial ones.
So eukaryotic tools on a bacterial-style blueprint.
Kind of, yeah. And even their control varies. Some have one origin, some have multiple, up to 4. But some archaea have another trick up their sleeve. Certain polyploid ones, like Halophorax volcanii, can actually replicate without dedicated origins.
Without origins? How does that even work?
They seem to use something called recombination-driven DNA replication, or RDR. It's usually a DNA repair mechanism.
Repair.
Yeah, if you have a break in your DNA, RDR can use another copy of the sequence as a template to restart replication. These archaea appear to have co-opted this repair process as a primary means of replication, especially when they have multiple copies of their genome. It's really quite remarkable. Oh, and they also have a unique polymerase, POL-D, alongside a eukaryotic-like POL-B.
Wow, okay. Archaea are definitely keeping things interesting. Let's shift gears to reading the blueprint. Transcription. The big eukaryotic feature is separation, right? Making RNA in the nucleus, making protein outside in the cytoplasm.
That separation is fundamental. It allows for an extra layer of processing and control that you just don't see in bacteria, where transcription and translation happen practically side by side.
And the eukaryotic genes themselves are structured differently too. Exons and introns.
Yes, most protein-coding genes are monocistronic one gene, one protein product. And they're discontinuous. You have the coding parts, the exons interrupted by non-coding stretches, the introns.
Which means the initial RNA copy, the pre-mRNA, isn't ready for translation yet. It needs editing.
Absolutely. It needs significant maturation. First, a special cap, a 7-methylguanosine, is added to the 5-foot end.
The front end, what's that for?
Protection. And it's a crucial signal for the ribosome later basically says, hey, I'm a valid message, translate me. Then, at the 3-bup end, the tail end, a long string of adenine bases is added, the poly-A tail.
Protection again?
Protection, yes, but also involved in export from the nucleus and translation efficiency. And in between capping and tailing comes the really intricate part, splicing.
Cutting out those non-coding introns.
Right. This is done by a massive molecular machine called the spliceosome, which is itself a complex of proteins and small RNA molecules. It recognizes the boundaries between introns and exons and snips out the introns, stitching the exons together.
And this isn't always done the same way for every gene, right? Alternative splicing.
Exactly. That's a huge source of protein diversity. By choosing different combinations of exons to include or exclude, a single gene can actually code for multiple different versions of a protein. It's how humans, with maybe only 20,000 or so genes, can produce such a vast variety of proteins.
Incredible efficiency. And getting transcription started in eukaryotes also involves more players than in bacteria.
Yes, it requires a whole suite of basal transcription factors. Things like TBP, the Tau-binding protein, which recognizes a specific DNA sequence in many promoters, and large coactivator complexes like mediator. They all assemble at the promoter to help recruit RNA polymerase II, the main mRNA-making enzyme.
Okay, so Archaea again, cytoplasm like bacteria, but eukaryotic like machinery.
You got it. Transcription happens in the cytoplasm. They can make polycistronic messages sometimes, multiple genes on one RNA, but they use a single RNA polymerase that looks a lot like eukaryotic Pol-2. And initiation. It's like a streamlined eukaryotic process, relying heavily on TBP and another factor called TFB to position the polymerase correctly.
So the theme continues. Simple setup, complex tools. Now, let's move to translation, actually building the protein. Elongation and termination are pretty similar across the board, you said.
The core mechanics, yes. But getting started, initiation, is again very different in eukaryotes. They don't just look for a Shine-Dalgarno sequence like bacteria.
They use that 5-foot cap we talked about.
Precisely. It's part of a scanning model. Initiation factors bind the 5-foot cap. Other proteins called PBPs bind to the 3-foot poly-A tail.
Wait, they bind both ends?
Yes, and they interact. This actually causes the mRNA molecule to form a loop, bringing the end close to the beginning.
Why loop the mRNA?
It's thought to boost efficiency and quality control. Once a ribosome finishes translating and falls off the end, it's already near the beginning, ready to start another round quickly. It also helps ensure only intact, complete messages get translated efficiently.
Okay, that makes sense. So the ribosome's small subunit, the 40S, binds near the cap.
The 40S subunit, along with the special initiator tRNA carrying methionine, forms a complex that binds the capped end and then scans along the mRNA until it finds the first AUG start codon. That's the signal for the large 60S subunit to join and start protein synthesis.
Now, once the protein chain is made, it has to fold into the right shape. Chaperones help with that everywhere, but what about organisms in really extreme conditions, like boiling temperatures?
Right. Protein folding is hard enough at normal temperatures. Imagine trying to fold correctly at 100 degrees Celsius or more.
Doesn't seem possible.
Well, Archaea, living in those conditions, the hyperthermophiles like Paradictium occultum, which grows best around 110 degrees C, have specialized chaperones. One key example is the thermosome.
Thermosome.
It's their version of the HSP60 chaperone complex. It helps other proteins fold correctly, and crucially, it works best at extremely high temperatures. Its ability to use ATP energy peaks around 100 degrees C. It's perfectly adapted.
Incredible adaptation. And finally, proteins need to get to the right place. Eukaryotes have the ER, Golgi, a whole internal trafficking system using vesicles.
Right, using the Sec61 channel to get into the ER or chloroplasts, then complex vesicle transport. Archaea, lacking these organelles, use systems more like bacteria, primarily the Sexic system, SEA, and sometimes the TAT system to move proteins across their cell membrane out into the environment or into the cell wall. Again, the proteins involved share ancestry with eukaryotic ones, but the cellular context is simpler.
Okay, last major topic, controlling all of this. Regulation. How do eukaryotes manage when and how much genes are transcribed? It seems like it could get really complicated with enhancers and silencers acting from far away.
It is incredibly sophisticated. We call it action at a distance. Activator proteins bind to DNA sequences called enhancers, which can be thousands of base pairs away from the actual gene promoter. Repressors bind to silencers, also potentially far away.
How can they influence the gene from so far?
The DNA loops around. The enhancer or silencer region, with its bound proteins, physically loops over to contact the transcription machinery assembled at the promoter, often interacting via that mediator complex we mentioned. This physical contact either stimulates or inhibits transcription initiation.
Like a genetic remote control, bending the DNA to make the connection.
That's a great way to put it. And eukaryotes also regulate access to the DNA itself by modifying the chromatin structure.
Changing how the DNA is packaged.
Exactly. Chromatin remodeling involves shifting or ejecting histones to expose DNA. Chromatin modification involves adding chemical tags to histones, things like acetylation, which usually loosens the chromatin and promotes transcription, or methylation, which can often lead to tighter packing and gene silencing.
So you can regulate by controlling access to the gene, not just the on-off switch directly. But there are boundaries, right? You mentioned insulators.
Critical. Insulators are DNA sequences that act like fences. They prevent an enhancer activating the wrong gene or stop silencing from spreading inappropriately along the chromosome. They define regulatory domains.
Okay, this really highlights the complexity. Now, this raises an important question. How do eukaryotes use tiny RNA molecules to fine-tune gene expression, sometimes even silencing genes completely?
RNA interference, or RNAi. A major layer of control. There are two main players: microRNAs, mRNAs, and small interfering RNAs, mRNAs.
How do mRNAs work?
mRNAs are typically encoded by the cell's own genome. They get processed by an enzyme called dicer into short, double-stranded RNAs. One strand then gets loaded into a protein complex called RISC, the RNA-induced silencing complex, which includes a key protein called argonote.
And RISC does the silencing.
Yes, the mRNA guides RISC to messenger RNAs that have a complementary sequence. Depending on how well they match, RISC can either block the ribosome from translating the mRNA or trigger the mRNA's destruction. It's post-transcriptional control messing with the message after it's made.
Okay, and RNAs, are they different?
RNAs often originate from external sources, like viral RNA or experimentally introduced RNA. They also get processed by dicer and loaded into RSC to silence target mRNAs, similar to mRNAs. But they have another trick.
Which is?
They can also guide a related complex called RITES, RNA-induced transcriptional silencing, back to the nucleus. RITES targets the corresponding DNA sequence on the chromosome and recruits enzymes that modify the chromatin, packing it tightly into heterochromatin.
So RNAs can shut down the gene before it even gets transcribed by changing the chromosome structure itself.
Exactly. It's transcriptional silencing, hitting the process at the source. Very powerful.
Wow. Okay, compared to that, Archaea must be simpler.
Generally, yes. Their regulation blends bacterial and eukaryotic strategies. Some archaeal regulators bind to operator sequences near promoters, much like bacterial repressors or activators. But others are distinctly eukaryotic-like, for instance. Some repressors work by binding directly to the TATA box, physically blocking the TBP transcription factor from landing there.
A direct block at the starting line. And their chromatin.
It's much simpler. They have histone-like proteins, but they organize smaller stretches of DNA, maybe around 30 base pairs, and they lack the complex pattern of modifications seen in eukaryotes. So chromatin modification isn't the major regulatory platform it is for us. It's, again, a unique blend.
So if we try to pull this all together, the big takeaway seems to be that Archaea really are this crucial evolutionary bridge. They have the simpler cell structure, maybe of bacteria, no nucleus, coupled transcription translation, but they run it using protein machinery that looks remarkably like the more complex eukaryotic versions.
That's the essence of it. And understanding that bridge is absolutely vital for projects like the bioplastics one we started with. To get those bacterial PHA genes working well in the eukaryotic diatom, P. tricornutum, scientists can't just drop them in.
They have to eukaryotize them.
Essentially, yes. They need to add strong eukaryotic promoter sequences, binding sites for the diatom's transcription factors, make sure the message will get the proper 5-foot cap and poly A tail, and importantly, if there are any sequences that look like introns, ensure they're either removed or won't cause splicing issues. It's about making the bacterial instructions legible to the eukaryotic machinery for high-level expression.
It really underscores how fundamental these differences are for biotechnology. Okay, for final thought then, maybe something for our listeners to chew on. You mentioned those polyploid archaea using RDR, potentially ditching replication origins.
Right, the recombination-driven replication.
If they found a way to replicate their circular genomes, even using a repair mechanism as their main strategy, what was the evolutionary pressure or advantage that absolutely required eukaryotes with their linear chromosomes to develop something as complex as telomerase? Why not just keep circular genomes or find an RDR-like fix?
That's a great question to ponder. What made linear chromosomes and telomerase the winning strategy for eukaryotes? Something to think about.